Okay. All right, I'm gonna start. Well, thanks. We just research on understanding the ecology and evolution, anatomy, microbial population, and using
That always happens to me. As soon as you start talking, everything's fine until you start.
Yes. I'm back. So it was just me, I guess, right? Okay.
Yes.
Oh, yeah. Well, yeah. So I was saying that to that end, he uses a combination of whole genome and metagenomic sequencing, population genetics, and computational biology and experimental approaches. He has made a significant contribution in 4 major areas. First, tracking pathogen transmission and evolution within patients, but also developing genome-wide association methodologies for studying microbial pathogens. And additionally, he has been characterizing diversity of microbiomes. Finally, he's also studying the evolution of bacterial pangenomes. And to date, he has published more than 100 articles, some of them in very prestigious journals such as Science, Cell, National Microbiology, or eLife. He has 5,500 citations. And lately, he has been co-leading the bioinformatics and computational biology model of the Canadian coronavirus variant rapid response network. Boy, that's a whopper. Where they are tracking SARS-CoV-2 evolution and transmissions in Canada. And today he's going to talk about the introduction of and spread of SARS-CoV-2 into the province of Quebec. Well, without further ado, Jesse, take it away.
All right, thank you so much, Santiago. Thank you for the invitation. I visited you at the CCG Cuernavaca. In the before times, it was before COVID, it was a few years ago. I had a great time. So meeting many of you, and I hope to come back in person soon. But for the moment, you have me virtually. Okay, so can you see my first slide?
Yep, yeah.
Okay, all right, excellent. So, right, so today I'm gonna be talking to you about evolution and transmission of SARS-CoV-2 using genomics. All right, so I'm actually, there are benefits and disadvantages of working in a very popular research area like the COVID pandemic. But the good news is that other people can do your work for you in your introductory slides. So there's a really nice viewpoint published in Science by Michael Martin, Dave Van Insberg, who parenthetically, I think, spent some time with you a few years ago during his PhD, and who's now in the lab of Katya Cole in Georgia. And in this paper, they outline basically 3 things you can do or you can learn from genomics. You can learn about pathogen emergence, so its origins, phylogeography, so geographic spread and introductions from one country to another, population dynamics, and adaptation. And I'm gonna tell you a story in 3 parts that touches on these things. So the emergence part, I'm gonna use as sort of just a little bit of background onto these methods of phylogenetics and epidemiology. Then I'm gonna tell you a story about phylogeography, about the first wave here in the province of Quebec, Canada, where I'm speaking to you from. Then I'm gonna talk about adaptation to non-human animals, so the spread and adaptation to animals like deer and mink and domestic animals. And finally, adaptation to avoid the human immune system, which of course we're interested in because that allows the pathogen to persist and continue in a hopefully post-pandemic world, but a world where there's a continuous cycles of evolution and evasion of immunity that allows this virus to continue to cause us problems. All right, so part 0, this is just some background. So emergence. So where did this virus come from? There are different theories, but I think that one in particular is really gaining more support. And there's evidence that comes from traditional epidemiology. Now, I'm not an epidemiologist, so this is a very hand-wavy sort of definition of what I mean by epidemiology, but I'm going to contrast it with genomic epidemiology using phylogenetics, which is really the kind of work that we do in my group. So this is just one example of, I think, a really beautiful traditional epidemiological study from Warraby et al. That's still a preprint. So this is very recent work that I encourage you to check out, a detailed analysis of the Huanan market in Wuhan, which they present evidence as really the epicenter of the emergence of SARS-CoV-2 from animals into humans. And what this figure shows is kind of a heat map. So this is just a floor plan of the market. And the coloring from gray to red is from surface swabbing and then PCR positive for viral samples. And so you can see that there's really a clustering around this part of the market. So they actually can geographically, at a very fine scale, refine where they think the virus might have hopped from animals into humans. And this is a photo of the animals that were in that location of the market that includes some raccoon dogs and some birds. So this is a really good candidate of where that spillover happened from animals to humans, okay? So this is also from their paper, and we all know what happened next, okay? So basically they're studying within the market. Those animals were probably not the ancestral reservoir. Probably it was a bat in an unknown location that made its way into some wild or farmed intermediate hosts, and then actually probably 2 spillover events within the market of 2 different lineages. So there already was a bit of genetic diversity in that viral population when it jumped over into humans. They did some spatial analysis showing that the cases in the city of Wuhan initially clustered right around the market. So this sort of local spatial spread throughout the city. And then with our globally connected world, we all know what happened next. And it's in every country in the world at this point. I think there are documented cases anywhere it's been measured. So that was some traditional epidemiology. Now, genome sequencing and data sharing, which I'm underlying, is the sharing of this data is really key and enables more people to look at the data and scrutinize it and make conclusions, and we arrive at better science faster. So the genome sequence was published in early January 2020. It was actually shared publicly on social media, on the internet, by these researchers before this Nature paper came out. Okay, so this is really a great example of data sharing. They did shotgun RNA sequencing from an infected patient from their bronchoalveolar fluid from the lungs. They didn't know what the pathogen was, so this is unbiased sequencing. This is a quote from the paper. They say, we generated over 56 million sequencing reads that were de novo assembled and screened for potential etiological agents. Of the 384,000 contigs assembled, the longest of about 30,000 nucleotides had a high abundance and was closely related to a bat SARS-like coronaviruses. They basically blasted it against the database and they find that it is a coronavirus. So this is what the genome looks like in terms of coding potential. As I said, it's about 30 kilobases and encodes 24 to 27 proteins, depending on how you count some different overlapping proteins. And it breaks down mainly into non-structural proteins towards the 5' end, and then the structural and accessory proteins towards the 3' end. The famous spike protein is the vaccine target. So that's in most vaccines, it decorates the surface of the viral particle. So in terms of nucleotide identity, this virus had never been found in nature before, at least not an identical relative. The closest match was from bats, so this is the most likely reservoir. But there's also some more distant relatives in pangolin, and then it's about 80% similar at the nucleotide level to the original SARS-1 that emerged and spread in the early 2000s, and then about 50% similar to the common cold. So a lot of coronaviruses are fairly harmless, but cause a cold. So this first genome was done with shotgun metagenomics. That's a very expensive way to go. It's a lot of sequencing. It's more than you need. It's labor intensive, but having the genome allowed research to design primers to do targeted sequencing. And as a result, we now have millions of SARS-CoV-2 genomes, most of which, or many of which are publicly, or millions that are publicly shared, I should say, which is really a phenomenal resource for research and what I base this talk on. These are mostly human swabs. So if you ever went in for a PCR test from a nasal swab, some of those will get sequenced in different countries. But some of these samples are from animals and wastewater. With that, I'll talk a little bit about animals, not so much about wastewater, but this is also a really interesting place to sample. Okay, also just making the distinction if we're doing deep sequencing, right? So typical Illumina sequencing, you're probably familiar with, you get very redundant sequencing. You get a whole bunch of Illumina reads if you sequence at a good enough depth that cover a given position in the genome. Okay, so this allows us to get a high quality consensus sequence, but then also if we really sequence deep enough, we can actually identify minor variants that are mutations that might have occurred within an individual patient, right? So some of these might be sequencing errors. We need to do some quality filters, but again, with enough sequencing, we can identify these mutations that happen within patients, as well as the consensus sequence, which is what we expect is the majority and what sort of gets transmitted most likely. Okay, so the first part of my talk is really going to just focus on these consensus sequence, just assuming that there's one clonal virus per patient. And at the end, I'm going to get into some of this within patient variation in the last part. All right, so a bit more about methods, phylogenetics and genomic epidemiology. We've learned that SARS-CoV-2 point mutations, so single nucleotide position changes, appear once every about 2 weeks. So this is the molecular clock, and it allows us to build time trees. So this is just a toy example. Imagine each of these dots is a sequence that's collected on different dates, and the branch lengths are proportional to the number of mutations that appear. Okay, so if you can plot this with time in weeks on the x-axis and the root to tip distance. So from the root, we have 1, 2, 3 mutations going from April 15th to May 30th or 31st. So there's a little bit of noise in our estimate, right? The clock is not perfect, but we can fit this clock. Here's an example. Here's a real example, and this is with colleagues as part of COVARNet, the Canadian Coronavirus Variants Research Rapid Response Network. It's easier to say COVARNet. And with the different variants of concern, alpha through Omicron in different colors. And so you can see that generally, there's a very nice correlation between the time and the divergence from the root and the number of point mutations. However, you'll notice, and you can really see it in bright red in Omicron here, where there's sort of a jump, right? So there's a jump in the divergence, and then it goes back to the typical slope. So I'm just gonna pose this as a question you can keep in mind about why that might be happening, particularly in these variants of concern, these sort of jumps that we see in the evolutionary rate. I'll come back to that a little bit. Okay, so now with that background about phylogenetics, about genome sequencing, this is all the background to bring us to my first story, which is about the phylogeography of the first pandemic wave in Quebec. So this is going back, rewind your head to early 2020, the early days of the pandemic. So this is a plot of essentially events that happened in Quebec, the province where I live, in early 2020. So in Canada, we mostly ignored the possibility of a pandemic and thought it would never come here and it would stay in China and it wouldn't spread until end of February, beginning of March, where it was clear that there were some cases. And then in gray, you can see the daily case counts really exploded over the month of March and into April. And a health emergency was declared mid-March. We had closures of borders and a lockdown, checkpoints at the borders. The Army was actually deployed in long-term care facilities. So for elderly people and people with special needs in Quebec, it really spread tragically. And actually the Army had to come in to help. Our first mask mandate was the middle of May. And then finally cases started to come down and some of these measures started to be eased towards the end of May, okay? So early on in the pandemic, I started asking, are we sequencing these viruses? Who's doing it? Can I help? And I got in touch with the Quebec Public Health Lab, colleagues at the McGill Genome Center, and quite early on, we started to Oh, sorry, okay. So can you hear me? Everything's okay? Okay.
Yeah.
Okay. Right, so we started to sequence nasal swabs from Quebec. And this is a zoom in just in the very beginning of March, and the blue are the sequences. And you can see that we're sequencing really a very good sampling, essentially the same number as the recorded cases. In some cases, we're actually sequencing a bit more than the number of recorded cases because the case counting was not quite accurate at that time, and the numbers were fairly low. Okay, so we do an especially good job of sampling early, and then it stays around 5 to 10% of the sequences throughout this first wave. All right, so the first question that I want to ask here is, how many times was the virus introduced into the province? And this was a question that was led by a postdoc in my lab, Dr. Carmen Leon-Morel, who now works as head of genomics at the Public Health Agency of Canada. So she's continuing this work in a broader Canadian context. So, and this was published late in 2021, where we have about 3,000 clinical samples from Quebec. And we put this in the context of 12,000 other sequences that were sampled globally. And what Carmen did is she built a big tree. The Quebec sequences are highlighted in blue. And their most recent common ancestor, I'll talk about this a bit later, is a little bit earlier. So that's in the sort of pink salmon color. So this is the same tree on the mirror image, and this is just highlighting 8 introduction events that are the most successful. So you can see it with the raw data with these dots. You can see that there's some parts of the tree that really have sequences that really persist forward into time. So these are very successful introduction events that are highlighted here. So of these are just 8 individual introduction events that give rise to over 50 descendants. So, and these account for over 50% of all the sequenced cases, okay? So out of 600 independent introduction events that we inferred on the tree, it's really only a minority that are very successful. So most go extinct, but some are successful. So here's another way of plotting this out quantitatively. So we can look at the transmission lineage size. So essentially the number of members of the descendants of that part of the tree. And here are these 8. The axis is not to scale, where these 8 have over, it's a little less than 8 that have over 100 descendants, right? But the vast majority of sequences, right? So this part of the distribution have 0 or 1. So they essentially go extinct. So this kind of distribution is over-dispersed. And this over-dispersion is consistent with super spreading events. That's something that, public health epidemiologists talk about a lot, where you get these events like a choir practice where everyone gets in, hundreds of people get infected all at once. This is something that we're actually seeing as a feature of the phylogenetic tree. We don't really know what these events are, but it's really evident from the tree that you do get these super spreading kind of dynamics. This is also evident in terms of geographic spread. So again, here on the x-axis is the transmission lineage size. So how many descendants does a lineage have on the tree? And the y-axis is a measure of the geographic spread within the province of Quebec. So Quebec is divided up into about 18 different health regions. And you can see there's a really nice linear relationship where bigger lineages spread to more regions. Okay, so these successful lineages have a lot of descendants, and those descendants spread widely geographically in the province. So the next question we can ask with this kind of data is not just how many introduction events, but where are they coming from? And this is a question that was asked by a new master's student in the lab, Sana Naderi, who used a method called ancestral state reconstruction. So very briefly, and I hope this is sort of intuitive, if you have a phylogenetic tree, and here the little rectangles at the tips are the observed states. So in this case, the states, in our case, is the province of Quebec or some other geographic location. Okay, so we observe that if a sequence came from Quebec, or here in this case, it's, let's say in black, if it came from another province or country. And in this example, we sort of infer the maximum likelihood, most likely ancestral state deeper in the tree, right? So we can infer that there was probably an introduction event into Quebec here where the ancestor was somewhere internationally. And then, there's going to be some uncertainty in the tree, and we can resolve this in different ways. Big caveat is this, of course, depends on sampling, is that if you haven't sampled from a given country, you would never be able to infer that a transmission event came from there. Okay, so there's big caveats there. So Sana applied these methods, and we could compare it. At least in the first wave, we had pretty good travel history for a subset of our samples. So about 300 people, their sequence was also associated with someone asked them where they traveled from. Okay, this doesn't happen so much anymore. And then for a larger subset, we can use the phylogeny using ancestral state reconstruction to infer where did the sequence probably come from. And then we can combine those two methods to sort of refine our estimate. So you can see that came a little bit from all over, but the thing that I want to highlight is that just based on travel history, there's 32% of people traveled from Latin America and the Caribbean. These are a lot of Quebecers. In the winter, they like to go to Mexico. They like to go to the Dominican Republic to escape the winter. So a lot of them were coming back from these locations. So that was their declared travel location. But if you just use the phylogeny, you see that that percentage drops down to just 9%. And you see that really the majority are coming from Europe or more from the United States. And this is probably having to do with, these are viruses that were sampled in Europe or the United States, maybe transiently went through Latin America, but they're really European or American lineages that were the ones that ended up in Quebec. So this just kind of shows how these methods can complement each other and give you a different picture of what's going on, but they're not totally discordant from each other. So the last thing that came out of this work that I wanted to mention is how the importance of timing. Okay, so there were lots of theories. People blamed spring break. So people had traveled and were coming back, and that's what started the pandemic in Quebec. That may have played some role, but there's a simpler explanation, which is founder effects. Essentially that the first viruses to arrive were the ones that tended to be more successful. So it's a bit of a noisy relationship, but you can see that when you plot transmission lineage size, so essentially how successful is a lineage on the phylogeny versus the time its most recent common ancestor when it was introduced, you see that the early arrivals, not always, but they do tend to be more successful. And some of these arrived even before the spring break period. Okay, so it's an easier explanation of founder effects. The first one to arrive has a better chance of spreading. And you can also see that some of these, just out of the circles, had no travel history. Okay, the triangles do have reported travel history, so you can see how these were things that were not captured at the border because there's limited capacity. These are cases that came in a cryptic way. We don't know where from if we were just asking about travel history. And I'm just showing here where mandatory quarantine for travelers re-entering Canada started here. So that could have also played a role in leveling off this curve and preventing transmission. So probably some combination of founder effects and these public health measures like quarantine made it so that by the end of March and April, it was really, really rare to see transmission lineages that gave rise to more than 10 sequenced cases. Okay, so with that little vignette, I'm going to talk about a recent project that we've been working on, which I'm excited. This is my first time talking about it, so I'm very, very pleased to. I'm sharing this with you for the first time in a public talk, which is looking at animal adaptation. So this is this cartoon from Warabi et al, and I'm just adding a next part of the story very roughly, right? So we have international spread, we have lots of humans infected, but then with so many humans infected, we have the ability for humans to infect animals, and then potentially for animals to infect humans. So we can start this zoonotic cycle, which potentially could be really bad, because if we get stable animal reservoirs that harbor the virus, it will make eradication of this virus, if it isn't hard enough already, effectively impossible, right? Because even if we eradicate it in humans, it's still in animals and it'll come back to us. The second reason is just whenever we give the virus an opportunity to evolve in different animals, it can be under different and unknown selective pressure. So you can select for different sets of mutations that might adapt them to the animal, but might have unknown phenotypes of virulence and transmission in humans. So it kind of opens up new evolutionary avenues for the virus. So there have been a number of individual studies on this. So this is just one example. So these are usually look at one species at a time. So this is an example from an experimental study where they experimentally infected ferrets and mink with the original SARS-CoV-2, and they selected for a bunch of, or 3 different mutations in spike, so this red variant virus, which they experimentally showed is not very good at infecting human airways. So there can be a trade-off where if you select to infect one animal, it might reduce the ability of the virus to infect humans. So that could be potentially a good thing, but it's something that we certainly want to know about. So they found some mutations, but these studies are mostly done, either experimental or sampling animals, but it's usually one animal at a time, relatively small studies. So they're kind of anecdotal and they don't really have a lot of statistical support for, what are the major mutations that are really associated with different animals. So we decided to have a comprehensive look at the data that is available to ask 2 main questions. So the first, how many transmission events are there from human to animal? Okay, because the virus is in humans, so it's going into animals. But then are there any secondary sort of spillbacks from these animals back into humans? So again, Sana used ancestral state reconstruction. So the same kind of idea of with the states being different countries or provinces, here the states are animal versus human, and how that state changes in the tree will tell us about these transmissions. The second thing she looked at is to ask, are there any animal-specific adaptive mutations? So are there any mutations that are really statistically associated with a particular animal compared to human? And to do this, she used a genome-wide association study. And this is a preprint that we just put up earlier this week, so it's really fresh. So I welcome all of your comments and questions about this. All right, so again, the importance of open data and big trees, right? So here we used available publicly shared genome sequence data. We didn't generate this data ourselves. So big thanks to everyone who's generating and sharing this data in GSAID. So that's the major database of consensus sequences for doing that. So Sana downloaded all of the animal-derived viral sequences from GSAID, so they were sampled from an animal host, and she included any species with over 30 sequences. So there's a few sort of exotic animals, but they're not very well represented. We want to do a statistically robust analysis, so we only looked for a reasonable sample size. And so this came down to mink is really, really well sampled, deer with just over 100 sequences, and cats and dogs with a more modest number of sequences, but we still can have a look. So what she did is, so we have these animal-derived sequence. She found the most closely related human-derived sequence. Okay, so that's its sort of most likely closest relative on a tree that maybe was involved in transmission, a recent transmission. She also added in, to fill out the tree, randomly sampled sequences over time. So she chose 50 sequences per month of the pandemic over the first 2 years. So that's a few thousand sequences. And she repeated this random resampling 10 times. So we get 10 sort of pseudo-replicate trees, so we can see how repeatable our analysis is. And for each of these trees, she built, I should say, each of these data sets, she built a maximum likelihood tree with 1,000 bootstraps. So kind of standard phylogenetic methods. This is what these trees look like. So these are just examples. Remember, we did 10 replicates of each of these trees. This is just one. Just like before, she did ancestral state reconstruction. I'm putting the caveat again, because this is very important. It depends on sampling, right? So this could change if we sampled a lot more cats and dogs, for example. Humans are pretty well sampled. So in light blue are human sequences. In the gold color are animals. And this is for cat, we're doing it separately for each animal. Okay, so for cats and dogs. And here, in this example, I'm showing we're looking for transmission events from animals back to humans. And you can see that we're identifying these using ancestral state reconstruction is these red upside down triangles, okay, that we identify a few, but we also did a bootstrap filter. So where we only are going to look at parts of the tree that have high confidence, okay, that have a high bootstrap support, and those are identified with blue squares, and you can see that there aren't any. Okay, so this is just one replicate. So sometimes there are, but you can see that especially in cats and dogs, we might identify these potential transmissions, but they're not very well supported on the tree. When you look at mink and deer, things start to look a little bit more interesting. Okay, so even with the blue, we see a lot of unfiltered transmissions, but it's a bit hard to read. But so you can see there's also some of these blue squares that are like very well supported parts of the tree where it was pretty clear that there was a mink ancestor that then gave rise to human sequences, and this is supported well with bootstraps. And you can see here's one example from deer. One little side note, which is noteworthy, is you can see that in particular on the deer tree, you can see these quite long branches leading to a deer sequence, right? So in these trees, I should have said this, the x-axis is scaled to genetic distance, so that it's proportional to the number of nucleotide substitutions that have occurred. So basically there's been like a lot of evolution happening in these deer branches, and that implies that there are according to the molecular clock, right, those went through multiple different hosts that we haven't sampled, but it suggests that there's quite a lot of deer-to-deer transmission, quite a lot of evolution is happening, but we're not sampling all the intermediate steps. Okay, so these trees are a bit hard to read. They're it gives you an idea of what we did, but this is just to be more quantitative to count the events. So this is a plot of the number of inferred transmissions from human to animal, okay? So yeah, in the human to animal direction for each of the species. And this is showing the 10 different replicate trees, okay? So we get a bit of an idea of the phylogenetic uncertainty. In the dark color are the bootstrap filtered. I'm gonna pay more attention to these. So you can see it's much more conservative. We see a pretty stable range of about 5 to 13 events where humans likely transmitted to animals. So it's pretty stable across all the different animals. When we don't filter for bootstraps, we get a lot more, but I'm a lot more confident in these. In the animal back to human direction, this is very rare. So those were the examples that I showed you in the trees. And you can see that for cat, dog, and deer, the estimates are 0, maybe okay. I showed you an example where you do see 1 in deer, right? So maybe it's 0 to 1 There's some uncertainty, but they're very, very rare, except for mink, where there's really been a lot of transmissions of animals back into humans. And this is consistent with what has been documented in some of the other papers on mink. But here we at least have it in a comparative perspective, so we can really see that, mink so far have been the major source of spill back into humans. And some of this can be due to sampling effects. Okay, so I'm going to keep highlighting that. So that was the first thing, looking at transmission. Next, we did a genome-wide association study to identify mutations that are associated with each animal. So we used a method called Poutine, which was named by a PhD student, Peter Chen, who just graduated, and he developed this method, which is in bioRxiv, and you can check out. It is not an acronym. It's just named after a favorite snack food here in Quebec that you should come and try, which is French fries with cheese and gravy, and it's really good in the winter, but has nothing to do with genome-wide association studies, other than maybe, just it's great and works really well. So what this method does conceptually is it focuses only on homoplasic mutations. So homoplasic mutations are illustrated in this tree by the X's. So these are mutations that happen multiple times independently in the tree. And this is important because those are statistically independent events. If you have a mutation that only occurs once in the tree, even if it then spreads through a lineage, those are not independent samples. So what this method is doing is a strong control for population structure. So you're controlling for the clonal tree structure, and you're looking just for these homoplasic mutations, which are independent mutations. And in the context of GWAS, we're looking not just for homoplasic mutations that are specifically associated with our particular phenotype in this case, which isn't really a phenotype, but it is the fact that that sequence was sampled from, for example, an animal of interest, in this example, a mink, okay? So I'm not going to get into all of the methodological details, but that's the essence of it. And we really like this approach, not just for viruses, but for bacteria, anything that has this really strong clonal population structure. So this is what the results look like. Not very interesting for cats or dogs. This is not too surprising because the sample size is pretty small. So we could have fairly low power to identify associations. So maybe in larger samples, we would find something. What these plots are showing, this is a typical, they're often called Manhattan plots, where this is looking along the viral genome. And the y-axis is the negative log p-value. So the higher the point, the higher statistical significance. In this case, there's nothing very interesting, but the idea is that if you see hits, they should look like skyscrapers on the Manhattan skyline. So these really tall, significant hits. In mink and deer, we do get some nice hits. Okay, so looking a little bit, I don't know, maybe not like a Manhattan skyline, but some significant hits that are genome-wide significant after correcting for multiple hypothesis testing. So you can see in mink, we've got 3 hits. These are all non-synonymous mutations, one of which occurs in the spike, 2 of which do not, but that have been previously documented in other studies. So this is some pretty nice overlap with existing knowledge, but here we're really providing rigorous statistical support for those associations. And what was really interesting, and that we're still trying to understand, is we get 26 significant hits in deer. And you can see by the colors, they're red and blue. These are both synonymous and non-synonymous mutations that are strongly statistically associated with deer. So none of these are in spike, so they're across the genome. So it shows the importance of considering potentially adaptive mutations, not just in spike, but across the genome. And even synonymous mutations could play a role in RNA stability and structure and regulation and things like that. So we're pretty excited about this, but still lots more work to do. So just to summarize this part, I keep stressing, with more sampling, we could reveal more transmission events and more GWAS hits. This is really based on the current state of available data. In mink, there might be limited uninterrupted mink-to-mink transmission, meaning there's more transmission of minks to humans and then possibly back to mink. So perhaps due to this limited uninterrupted transmission, perhaps there's less opportunity for adaptive evolution. So that could explain the relatively small number of GWAS hits, if we can use that as a measure of adaptive evolution. Whereas in deer, there's evidence for more sustained deer-to-deer transmission. So there's not a lot of cycles of deer to human to deer. It seems to go into deer and it spreads pretty widely, and there's different lines of evidence to show that. And maybe that could explain why there are more potentially adaptive mutations in deer, because the virus is circulating, has just larger sustained population sizes in deer. These are both hypotheses because, of course, these GWAS hits, right? So these are statistical associations with each of these animals, and they're candidates for experimental follow-up. So we really don't know why, in most cases, these mutations are associated with deer. Are they adaptive? Do they have trade-offs for infecting humans, right? So this is also really interesting. Or is the virus really going down an evolutionary pathway to be really good at infecting deer, which means that it won't be good at infecting humans? But that's not necessarily the case, right? So these are the kind of follow-up questions that would be really interesting. And of course, the big question I think everyone wants to know is, could the next variant of concern, like Omicron, come from an animal reservoir? I think it certainly could. We have not found that yet, but I think it's important to continue these investigations because it could. Okay, part 3, this is the last part. I'll spend the last 10 minutes or so and hopefully leave some time for questions, looking at adaptation to host immunity. All right, this is another piece of collaborative work, another recent preprint, not as recent as this week, but pretty recent, that was led by a master's student in the lab, Afno Nguyen, who's now a PhD student in Toronto. And this was work that was led by 2 immunologist colleagues at McGill, Jorg Fritz and Ciro Piccirillo. And Jorg and Ciro did the experimental part of the study, which I'll explain pretty quickly, but it was to do fine scale antibody epitope mapping. So they're looking for what parts of the SARS-CoV-2 proteome, so all the proteins that it encodes, are targeted by the human immune system, meaning are targeted by antibodies. So this is using a peptide array, a synthetic array, where they have a peptide library printed on a chip. So this is each a little 15 amino acid segment of the genome on a chip that can then be queried, kind of like a microarray, by serum from either infected patients or controls, and with a fluorescent assay to see if there are antibodies that bind. So you get a profile across the genome of where antibodies are binding. So this allowed us to query almost 4,000 unique epitopes. So this, that by epitope is, it's a 15 amino acid segment of a protein. And this covers most of the major open reading frames in SARS-CoV-2. It's not quite the whole genome, but it's pretty close. And this was done on 15 patients. Some controls, some infected. I'm not going to get into that. It's in the preprint, but it's actually not a major source of variation. So I'm just going to define epitope sites that are sort of bound in general above a threshold. So the question that we really wanted to ask is SARS-CoV-2 under selection for immune escape within patients? And it seems intuitive that, of course, it should be under selection, but it's not guaranteed. And in fact, in influenza, it's not really the case. And the reason for this is that transmission probably occurs before there is much selection from the immune system. So this is an example from an article looking at flu. And you can see that the viral load in red peaks before adaptive immunity, that is the antibody response, really kicks in. So most of the transmission happens when viral loads are high, you're shedding a lot of virus. And so the virus will continue to transmit and evolve really before it gets under any appreciable selection to evade adaptive immunity. So there's not a lot of selection within an individual host. It happens more at the level of the transmission bottleneck. This is some data to support this. This is analysis from Catherine Zhu and Jesse Bloom, where they looked at genome sequences from influenza. And again, similar to our epitope mapping, they have antigenic and non-antigenic sites. So these are equivalently basically like epitopes or non-epitopes that antibodies bind to. And they're looking at the within host level and the global level. So this again is the distinction I made at the beginning of my talk, where with deep sequencing, you can look at mutations that are happening actually within an individual patient. So at the within host level, and the global level is really at the level of those consensus sequences and sort of the big phylogenetic trees that kind of show us transmission, okay? And so they break down the evolutionary rate according to the mutation type. So either synonymous, non-synonymous, or stop codons, right? So non-synonymous mutations change the protein sequence, synonymous do not. And of course, stop codons are very damaging, right? They stop the protein entirely. And as you can see in blue triangles, they're in general pretty rare, right? Because they're deleterious. So in this analysis, synonymous sites, so in orange, are selectively relatively neutral. So they tend to evolve at the highest rates. So they're kind of unconstrained by evolution. They just accept mutations. But non-synonymous mutations are generally rare because most of them are deleterious. So they break something in the protein and they're removed by purifying or negative selection, right? So they tend to be more rare. The exception, of course, is this column, is in antigenic sites. Selection appears to favor non-synonymous mutations at the global scale. So at the global scale, we have a really high rate of non-synonymous mutations, but we don't really see this within individual hosts. So this supports the model that in flu, we do have high rates of evolution. So that could be due to immune escape, so selection to avoid the antigenic response or the antibody response, but this really happens at the global level, but not within hosts. Okay, so we basically copied this. Okay, we did something very similar for SARS-CoV-2 on the left at the within host level, on the right at the transmission level, meaning looking at consensus sequence. Okay. And so this is broken down by the 6 major coding regions of the SARS-CoV-2 genome. And then in orange is whether it's an epitope site in that gene, and gray if it is not an epitope site, okay? You can see that in terms of within host evolutionary rate, it's pretty similar within and between epitope sites. The blue arrows are showing that it's actually, the rate is actually slightly lower in epitope sites. Okay, so there's actually some evolutionary constraint, not very noticeable. Okay, in the epitope sites. However, at the transmission level, okay, so over longer evolutionary timescales, we see particularly in spike and really strongly in the nucleocapsid, the N protein, that there's really a faster evolutionary rate in epitope sites compared to non-epitope sites. So the conclusion here is actually pretty concordant with what has been found in influenza, is that mutations in epitopes that are targeted by antibodies tend to accumulate at the level of transmission, but not really within patients. Okay, so this is consistent with most of the selection really happening at that transmission bottleneck and not within patients. And this, again, I should say, is based on about 40,000 sequences publicly available in NCBI. Here we had to go to NCBI instead of GSAID because GSAID just has consensus sequences, but NCBI has the raw reads. So we need the raw reads to do this within host analysis. Same analysis, but here we're looking at the ratio of non-synonymous to synonymous mutations. So another measure of selective constraint. So both within, so polymorphic, non-synonymous and synonymous mutations within patients and then between patients. And again, you can see here, actually, you can see that in some cases, these red arrows shows that there's a little bit of a higher N/S ratio in epitope sites in the spike and the nucleocapsid. So maybe there is a little bit of selection for immune evasion happening within patients, but it's very small in comparison to what you see upon transmission, where you see really, really high DNDS ratios in S and N, and effectively negligible DNDS in non-epitope sites. Okay, so again, this suggests that positive selection is stronger between patients. So at the transmission level, particularly in the spike and the nucleocapsid protein in epitope sites. And this was all looking at first wave data, genome sequences from the first wave, so January to July 2020, but we see similar patterns in wave 2. Also comparing the waves, so first of all, we identify thousands of single nucleotide variants in epitopes. So most are identified in wave 1. A smaller number are unique to wave 2, and there's a fair amount of overlap. But what's notable is that these are initially rare. Okay, so the y-axis here is just the relative abundance. So how frequent do we see each of those mutations? So in wave 1 in blue, they tend to be pretty rare. But then ones that are in wave 2 or that are present or persistent in both waves are more prevalent. So this suggests they start off rare, but then they increase in frequency during wave 2, which is consistent with a sort of population, global, all the sequences in NCBI, general increased selective pressure for immune evasion to avoid those non-synonymous mutations in epitope sites. We also compared variants of concern, right? So we have alpha, beta, gamma, et cetera. We actually looked at both variants of concern and variants under investigation. And we find that, perhaps unsurprisingly, they're quite enriched in, again, non-synonymous mutations in these epitope sites compared to any other lineages that are kind of down here, have a median of around 4 mutations in epitope sites, but the variants of concern are, up here closer to 10. So this, again, suggests that maybe some of the selection pressure for variants of concern is to evade immunity, or it could be at least a secondary effect. Maybe they're selected for transmission and then just by accident, they also evade immunity, but they certainly have a lot of these mutations. Here are the different variants of concern ranked. And you might not be surprised, Omicron, the current dominant variant, really is the winner in having over 30 non-synonymous mutations, signature mutations that really define that lineage to the exclusion of others. Really, a lot of those are in epitope sites. It has a lot more than other variants. It explains a lot of its immune escape. A lot of these are in spike, unsurprisingly, but the flip side of the story is, there are a fair number in other parts of the genome as well, and I think these are not to be ignored. I think there's a lot of focus on spike. It's a great thing to focus on for vaccines and so on, but the rest of the genome also is probably targeted not insignificantly by selection for immune evasion. All right, so to wrap up, so just for this part of the talk, so pretty similar to influenza, SARS-CoV-2 is mostly under purifying selection within patients, just removing deleterious mutations, but not so much selection for immune escape. But positive selection occurs for antigenic escape, really at the level of transmission between patients. And this makes sense also with the viral load peaking before the antibody immune response kicks in. So lots of future questions. I'm not going to list them all, but I should say that all of this data from NCBI, right, those are sort of the average infection, which tend to be mild, that tend to be acute, probably don't sample a lot of long-term selection in chronic infections. So this could be very important. You actually could get very different patterns of a long-term infection. There probably is. There's a lot of time for the virus to interact with the immune system. So that's probably an exception, but it's probably an important selection. An important exception. And similarly, animal reservoirs weren't really included in that analysis too. And again, those can be very important, but maybe should be considered sort of separately. Okay, so with that, I'm going to wrap up. I'm going to thank my group, a lot of great collaborators at the Quebec Public Health Lab, other collaborating labs at McGill, the McGill Genome Center, who did a lot of the sequencing and bioinformatics support in Quebec, members of my lab, who I've mostly thanked individually already. This is our lab barbecue in the pre-pandemic times. There's new people in the lab. Some of you will recognize Dr. Olga Perez-Carrascal, who came to my lab from you and has now gone on to another postdoc in Berkeley, but this was a picture before she left my lab. So that's it. Just a pitch. There are some positions available in my lab if you're interested, mainly in computational biology of SARS-CoV-2 genomes. There are other projects as well. Feel free to contact me if you're a PhD student who's graduating and looking for an opportunity. I was really happy to recruit Olga, and I'd love to have someone else join me from Mexico to make more links. All right, so with that, if there's time for some questions, I'd love to take them. So thanks so much for your attention.
Thanks very much, Jesse. Very interesting talk. Questions, anyone? Come on, don't be shy.
Well, I want to ask something about this. I think it's a really impressive work on subcontext. This is the first time that I see some population genetic principles applied to the epidemiology as several aspects, adaptation and evolution of SARS-CoV. I think that some of the problems, I think that the variants of concern contain multiple mutations, several mutations. I'm not, this is correct, but my question is about the role of recombination in the possibility of co-infection of the SARS-CoV variants in the same patients, or even in the reservoirs in the field, etc. If recombination has a role generating new mutations, if it explains some of the bumps observed in the first graph, that's my question. If the recombination has a role in the diversification and the origin of new variants.
Yeah, that's a great question. So I didn't talk about recombination at all. Most of the analyses that we did, like most of this data is from the stage of the pandemic before there were appreciable numbers of recombinants that were documented. More recently, there have started to be recombinants documented, which basically takes two, like you said, it takes a co-infection, and well, I should say, recombination can happen but never be documented if you just don't have genetic diversity. Because, you know, trivially, if a recombination event occurs between two identical viruses, then you can never detect it, right? So you need enough mutations to essentially tag the two parents. And so I believe the documented recombinants are between Delta and Omicron, which are different by 30 or 50 mutations. And of course, they need to co-infect, so they need to be co-circulating. And those conditions were right in Europe. Actually, there was a documented paper from the Americas in the US and Mexico, which I think were two different sublineages of, no, is it Alpha and Delta? I forget. In any case, all this to say, there have been a few documented. So far, none of them has been particularly successful. So they've maybe persisted, but they haven't outcompeted. So it hasn't given them, a particular jump in the fitness landscape. But it definitely can happen, and I think that it's something to look out for sure in next variants of concern.
Thank you very much.
I see just a technical question about what software did we use for ancestral state reconstruction. So we used the APE packet, the ACE script, or the ACE. APE is the package in R, and it's the ACE method that's implemented in there. So we use both maximum likelihood and parsimony, and we sort of compared the results. So you can, the details are in the preprint. Yeah, so it's a fairly standard method. Again, I was very open about sensitivity to sampling. I think there are other Bayesian methods that will try to account more for the sampling process. These are a lot more complicated, so we went with something that was relatively simple. But I think that in the future, I think that there are more refined methods that could help account for some of those different sampling processes.
Yeah, go ahead, please.
Thank you. A very nice talk. I don't know why my camera is not getting on. I'm sorry. Anyway, great to hear you. My question, Jesse, is how strong is do you think is the impact that I guess most of the samples you have access to are throat swabs regarding your analysis on within individual evolution? If it's all a swab, a throat swabs, what you get, these are all very recent infections. So there's hardly any possibility, I would guess, to really see evolution and immune escape, etc. On those samples. Is it possible to filter your samples in order to find out others that are perhaps from other tissues or organs deeper in the body, where you probably would have greater chances to find signals of selection? What can you comment on that, please?
That's a great suggestion, and I think that those samples are rare, and there are a few focused studies mainly on chronic infections where they're being sampled over time. I think even that it's still mainly those swabs. I think the alternative is the bronchoalveolar lavage, where they basically get a lot of fluid from the lungs, so it's a bigger population, different tissue. It's a great suggestion. It would be I'm not, again, our approach was sort of really just to look at all the data, see what we can download from NCBI. So as I said, it's just like a sampling of what is available. It's like the 1,000-foot view. But no, it would be very interesting to filter in that way. I'm not sure if we could. It might take some manual curation, but that would be a great suggestion.
And that probably would be very advisable to with our collaborators, or find the people who are actually sampling, to try, and those in working in hospitals, to really try to get samples from not just the throat swabbings or in the nostrils, but deeper in the tissues of those that have long COVID, etc., to really understand these problems that after we are getting into the third year of the pandemic, to really be better prepared to eventually treat and understand how this evolves in our bodies. Thank you very much for your comments. Yes.
Yeah, absolutely. Great questions.
So there is one question in the chat, Jesse, about residual water variants, and that they might not be traced to human host, I guess is the meaning.
Yes, I've seen those. I've seen, actually, there's a recent preprint that came out, which I need to read. I haven't seen that data in Canada. I am collaborating on some wastewater work as well, so I will keep that in mind. But it's certainly interesting, and I know that there's an initial paper from New York City, right, that postulated that some of the variants in wastewater were maybe attributed to rats. Like rodents of some kind. That might be rodent that's wastewater sampling some of these animal populations, which could be interesting. In actually just a Twitter exchange with John Dennehy, who's in New York, who's doing some of the sampling. None of the animal associated mutations that we identified are present in the sort of cryptic lineages that they found across the US. So I think he thinks that it's not only animals. It could be there could be other reasons for some of these cryptic lineages that are hard to trace. So I don't know. It's a great question, and I think it's something to keep studying.
Any more questions? So I have one, Jesse. Well, actually I have two, but I'm gonna start with the first one. So it was very interesting to see that basically you find positive selection on the epitopes at the transmission level, right? But that was between humans. So what about looking at transmission with other species? Because there you have a complete different adaptive landscape, so to speak, right? Well, not completely different, but at least slightly different than if you were to look within the human population.
Yeah, that's right. And I imagine, I'm trying to think if there are, epitope maps for mice as well. I imagine that exists, right? So for different model animals, I do think that that's something. So I'm collaborating with also part of CoVarNet, and it's a very cool initiative in that I'm working in the computational biology part, but then there's a whole pillar of people, including Selena Sagan, who's my collaborator on the animal part, but people like Angela Rasmussen, people who are doing animal experiments and things like that. So I think they might have that kind of data, trying to actually map the epitopes for these different animals. Anecdotally, one of the hits, and I didn't really get into all of the details of the hits, but one of the hits for mink that we identified that's been found previously is N501T. So it's position 501 of the spike. And that's an interesting one because that was also a mutation that happened in the 3 first variants of concern. So alpha, beta, and gamma they all had N501Y. So it's sort of an interesting thing where it seems that like Y seems to be adaptive, and that seems to be mainly selected, I think, for binding the receptor. So it's maybe it has some immune effects, but I guess it's mainly at the level of the receptor. But for mink, it seems to go to a T, right? So it's a subtle difference. Anyway, I think that is mainly at the level of like receptor binding, but there for sure could be similar things happening at the level of immunity, or different animals that just have less, overall less of an antibody response. This is, a hypothesis to explain why bats are the reservoir of lots of viruses, that they generally just have, I think it's called immune tolerance, where they sort of just don't have a very strong immune response and they manage to survive, but they kind of just let the virus do their thing and they don't have a lot of inflammation and they're kind of okay. That's a simplification, right? But I think that a lot of this.
Right. And yeah, I mean, along those lines, and in the context of all the One Health thingy, so do you think, I mean, given all the things that you now know about SARS-CoV-2, so basically you can have like several, but let's stick to 2 scenarios. Basically, you can have like for so many bacterial pathogens where you have populations that are species specific for a given species. Like that seems to be the case, for instance, for Acinetobacter baumannii. So what do you think is going to be for SARS-CoV-2? So do you think it's going to be like separate populations that every now and then they have transmission between them, but on the whole, they seem to be well differentiated from one another?
Yeah, I mean, that's the big question, right? And I think that in general, at a family level, coronaviruses tend to be pretty flexible about infecting hosts, where it seems, I mean, SARS-CoV-2 was able to jump into humans and be pretty successful even immediately, before all this evolution. They were capable of infecting humans. So this sort of generalist strategy, where I think so I mean, I don't have a great answer, but so I guess what I'm saying is, I think it's plausible that, say, just to use the example of deer, where it seems like it's spreading a lot and there's deer to deer transmission, and plausibly there'll be adaptation to deer, right? So it'll sort of branch off and go in its own direction. But I guess just based on the sort of bigger evolutionary tree of coronaviruses, where you can see they jump from bat to pangolin to humans, that I don't imagine that say, deer population, for example, would be totally isolated. I think it would take a lot, or maybe be impossible, for it to not be able to infect.
Gotcha.
So I think that's the challenge, because I think that's where evolution becomes kind of unpredictable. Is, if you think of a fitness landscape where you have a peak for humans, and then there's a higher peak, but then that they're not connected, but maybe it's connected through deer, because you have to go through like a path that's not adapted for humans, and you accumulate all these mutations, but then once you've accumulated them, it's like really adaptive for humans. So I actually, so I think it's plausible the virus can evolve in a like totally different direction in some animals, but then still infect humans, and maybe with, you know, even higher transmission or you know, other unintended consequences. So I think it's something to look at. Yeah.
All right. And there is another question.
Yeah, fecal samples aren't
Right.
yeah, I mean, that is something, especially in wastewater, you know, a lot of what is sampled is probably what is shed through the fecal route. So I also wonder if, you know, maybe some of those cryptic lineages are things that are selected like in the gut and then are shed, but I agree it's not really collected. So I think be very interesting, but that's something that's like totally missing from, you know, at least the data sets that are readily reported. So I think that would be a cool study.
All right. I guess we don't have any more questions. Also, thanks very much, Jesse. It was an amazing talk.
My pleasure. Thanks so much for inviting me. And as I said, next time I'll come visit you in person.
Right. By all means.
It'd be great to see everybody.
Okay.
Thank you very much.
All right. Thanks very much.